Dear students, we had spoken about immunoprophylaxis in the previous session. You know what is it? It is a prophylactic measure or a disease preventing measure by improvising your immunity. So, and we learned that the use of vaccines are the technique that we can use to improve our immunity. Now, what are the different types of vaccines? That's what we are going to talk about in this. Basically, we have seen different types of vaccines: the live attenuated vaccines, the killed vaccines, the toxoid vaccines. Then we had also seen the recombinant vaccines, subunit vaccines in the previous sessions. So let's go into detail into the main vaccines. The first one, live attenuated vaccines. Here, microorganisms are attenuated or disabled so that they lose their ability to cause a significant disease. But at the same time, they retain the capacity for transient growth within the inoculated host. And some agents are naturally attenuated by virtue of their inability to cause the disease in a given host, although they can immunize these individuals. And the first vaccine which was used by Jenner is of this type, because vaccinia virus, when it was been, that is a cowpox causing vaccinia virus, when it was being inoculated into humans, it confers immunity to smallpox but do not cause smallpox. So that was a case of natural attenuation in which, by inoculating into another host, what is it? The organism or the microbe was getting attenuated. Now, this can also be achieved by growing a pathogenic bacteria or a virus for prolonged period times under abnormal culture conditions. And this selects the mutants that are better suited for growth in the abnormal culture conditions than in the natural host. Now, for example, in the case of an Mycobacterium bovis, what happens? An attenuated strain of Mycobacterium bovis called the Bacillus calmette-guerin vaccine was developed by growing M. bovis on a medium containing increasing concentrations of biles. And after 13 years, this strain had adapted to grow in strong bile and had become sufficiently attenuated that it was suitable for a vaccine against tuberculosis. So, but of course, thus was the BCG vaccine which was being developed. But in US, of course, there are some because of the variable effectiveness and difficulties. What happens? BCG is not used in US. Okay. So the Sabin form of polio vaccine and the measles vaccines, they also are some examples of the attenuated vaccines. So in the case of live attenuated vaccines, you take the bacteria or the virus which you have, and then you make them attenuated. What is attenuated? That is, you make it in a situation that it loses its virulence, but it loses its virulence, or it has no virulence but it has a immunogenicity. That is, once it has been inoculated into the into a host, what happens? The immune response of the person is being triggered. So the organism will be living, but it will be attenuated. And how to attain this attenuation? By growing it in an abnormal culture condition so that it will not be able to cause a virulence. Okay, and some of these vaccines that which we have developed like that is a vaccinia, the smallpox virus, the smallpox vaccine, measles, mumps, rubella, or the MMR vaccine, which has been given as a combined vaccine. Then varicella for the chicken pox, influenza, which we have for the nasal sprays and all that. Then you have yellow fever virus, zoster virus, and all that. Okay, these are all live attenuated virus vaccines. Now, how to attenuate? We had already mentioned in the case of bacteria. In the case of virus, you can do this by introducing it into a species in which it does not replicate well, such as a different cell culture or an animal embryo or a live animal in which the virus cannot multiply rapidly. And the initial virus is applied to the host cells in a series, and after each passage, what happens? The virus acquires or possesses a mutation, and it becomes better at replicating in the host cells, but with that it also loses the ability to replicate in the human cells. So basically, in attenuation, we try to make the bacteria or the virus incapable of what? Causing its a disease, but at the same time it will be what? It will be immunogenic for the person. Now, what are the advantages of this one? It activates all the phases of the immune system. Even the IgA is been, IgA is the last type of local antibody which has been produced. Even that has been produced, and it provides a long lasting immunity, and boosters are required less frequently in the case of live attenuated vaccines, and it is of low cost, and it gives us quick immunity, and some of them are even easy to transport and administer. And the vaccines also have a strong beneficial non-specific effects, that is effects which go beyond the specific protective effects against the target organisms. Okay, now coming to the disadvantages. They also have some disadvantages. Now, in extremely rare cases, what happens? Natural mutations can cause a reversion to virulence. So in this case, the virus can revert to a wild type or develop into an entirely new strain after the process of attenuation. And so there are chances of reinfection because the virus is being found to be live, but it has lost its virulence, but sometimes some mutations can happen to it and it might revert back to its wild strain. Then live vaccines are not usually recommended for immunocompromised patients because they have a risk of potentially severe complications to occur. And some of the live strains typically require advanced maintenance such as refrigeration and fresh media, making transport to remote areas very difficult and costly. These are rare instances. So now let's go into the next type of vaccine, the killed vaccine. Sometimes what happens, when it is not safe to give a live vaccine, you can use a killed vaccine. This vaccine is been created mainly by the treatment with either heat or the chemicals, and it is critically important to know that you should maintain the structure of the epitopes on the surface antigens during the inactivation. Okay, heat inactivation can be done, but it is often unsatisfactory because it causes the extensive denaturation of proteins, and what happens is many of the epitopes on the higher order of the protein structure are likely to be altered significantly. So you can kill a bacteria by two different methods, either by heating or by chemicals. When you heat it, what happens? The chance of protein denaturation at a complete and an extensive level is very high. So what happens? You always prefer a chemical inactivation. Now this can be done by using either formaldehyde or various other alkylating agents, and the Salk polio vaccine, okay, this is the inactivated polio vaccine is an example of this by formaldehyde inactivation. Then the hepatitis A virus vaccine as well as the rabies vaccines are examples of killed vaccines. Now it has a negative disadvantage here. It may not always induce an immune response at the first dose, and hence what happens, you will have to give various doses of the vaccine, and the response may also not be long lived. And compared to the live vaccines, they have a less strong immune response compared to the live vaccines in the case of the killed vaccines. And coming to the safety and stability, it has been found to be more stable than the live attenuated vaccines, and they have no live components, and so there is no risk of any, including having the disease, having the chance of inducing the disease is in fact nil in the case of the killed vaccines, and they are more safer and stable than the live attenuated vaccines. Then the next vaccine that we have is the subunit and the conjugate vaccine. Now sometimes some subunits, okay, the antigenic portions of a microbe can be purified and they can be used as a vaccine. That is called a subunit vaccine, and it can be either for example, it can be any carbohydrate based or protein based component, or even it can be a conjugate based also. Okay, so in the case of subunit vaccine, what is it? You take a microbe, you take either its protein content or its carbohydrate content, and then you use it as a vaccine. Now these vaccines, since they do not contain any living components, okay, and the chances of inducing a disease is been found to be very less here. They only contain the antigenic parts of the pathogen. Okay. Now, vaccines composed of bacterial polysaccharide antigens. They are used in the case of pneumococcus as well as Haemophilus influenza. But it has been found that these polysaccharides, which have been used against pneumococcus as well as Haemophilus influenza, they are T-independent antigens, and they tend to elicit only a low affinity antibody response and poorly immunogenic in infants. So what happens is, sometimes to improvise the immunogenicity of these subunit vaccines, you might take those polysaccharide antigens and couple them with some proteins to form the conjugate vaccines. So when you take a subunit vaccine and conjugate it with the protein, you form a conjugate vaccine. Okay, and these vaccines will help to elicit the helper T cells to stimulate the germinal center reactions, and as a result you get a better response, a better T and a B cell response when you give a conjugate vaccine. So the difference you should remember, the subunit vaccine is only the you are only taking the antigen. Now it can either be protein or carbohydrate that is there, but to increase its immunogenicity, sometimes it might be combined with some other proteins, so then it becomes a conjugate vaccine. Now the examples that we have here is the hepatitis B or the influenza vaccine, the Haemophilus influenza type B or the Hib vaccine, the pertussis in the case of DPT, and the pneumococcal, meningococcal as well as the human papilloma virus. These are all treated with the help of subunit as well as the conjugate vaccines. Now for subunit vaccines, a specific protein from a pathogen is isolated and then it is presented as an antigen to activate the immune response. Now another way that we can take this is, sometimes what happens, you use the technique of genetic engineering. For example, a gene which has been coding for that particular protein is been inserted into a virus or cells, and after the virus production or cell metabolizing is being done, the virus protein is created and it is recognized by the immune system and it will give further protection. So subunit vaccines are also known as recombinant vaccines sometimes, because the proteins or the subunits are being generated by recombinant DNA technology. Hope so this is clear to you. We will continue in the next sessions. Thank you.